Hi everyone, welcome to the second video of the RNA-seq for Beginners series. In this series, we'll be walking through key concepts and tools to analyze RNA sequencing data effectively. Whether you're new to RNA sequencing or looking for a chance to enhance your analysis skills, you're in the right place. In today's video, we'll be covering Gene Set Enrichment Analysis. This is a powerful method to interpret RNA sequencing data and understand biological significance behind gene expression changes. We'll break down what GSEA is, why it matters, and how it helps uncover meaningful pathways in your data. If you haven't watched the first video in our series, be sure to check it out for a solid foundation on RNA-seq analysis basics using DE-seq2. We're actually continuing on the same code that we used in that video, so if you haven't seen it, go check it out and then come back to this video. Otherwise, let's get started and explore GSEA. So first, you may be asking, what is Gene Set Enrichment Analysis? So Gene Set Enrichment Analysis is a method that helps us understand which biological pathways or gene sets are changing in an RNA sequencing experiment. This is in comparison to just focusing on individual genes. So the way we do this is we start by ranking all the genes based on some sort of metric. Usually this is log two-fold change, and in the previous video we calculated a log two-fold change for each gene using DE-seq2. Then we take a predefined gene set, like a specific pathway, that has a list of predefined genes, and GSEA will then check if this gene set tends to cluster at the top or the bottom of that ranked list set we generated using DE-seq2. So you may be asking, what are the outputs of GSEA? How do you interpret these? So the outputs of GSEA are really three main numbers. The first is the enrichment score. The second is a normalized enrichment score, and the third is an adjusted p-value. Starting from the top, the enrichment score measures how concentrated that gene set is towards one end of this ranked list. It's like a running sum that spikes when a gene from the set appears. But the gene sets vary in size and distributions, so we have to normalize these enrichment scores using many random permutations, typically for 1,000. And this is really just normalizing on gene set size and general noisiness of your RNA sequencing data set. This will give us a normalized enrichment score, and there will be an associated adjusted p-value for each of these NES scores. And what our lab typically does is any pathway that has an NES score greater than 2 or less than -2 and has a p-adjusted value of less than 0.05, this usually indicates something that's actually really biologically significant. So you may be wondering, where do you get these predefined gene set lists? Well, that's what we'll be doing next. So here we will be downloading the predefined gene sets that we'll use in the GSEA analysis. Typically I use a gene set list called the Hallmarks gene set as my first pass through an RNA sequencing experiment. And the way we download this is by going to the MSigDB website. And I just type in GSEA here. And then we have to explore the MSigDB collection. We come here to the Hallmark gene sets. Scroll up here, there's 50 gene sets within this Hallmark data set. And we want the gene symbols because that is how we annotated our RNA-seq data. I've already entered my email, but they may prompt you to enter your email so you know who's downloading these files. But ultimately, this is what downloads, and that is what we will be reading into R to perform GSEA analysis. I just want to give a quick disclaimer before we jump into the code. The requirements for this particular analysis will be R version 4.2.2, that's what I'm using today, and here are the 4 packages that I have installed into my R environment so that I can perform all the necessary analyses. So let's get into the coding. So like I said, we're continuing on from the DE-seq 2 step-by-step guide, and this is where we left off. And we will be taking the output from that tutorial and then plotting the GSEA analysis, as well as creating a volcano plot where we take one specific gene set, in particular the G2M hallmark gene set, and overlay that on the volcano plot to see what it looks like on top of all of the RNA-seq data. So, if you don't remember, we started with this results file called df. And what we actually need to do is rename or move the row names to a column. This just facilitates downstream analysis. So what we'll do is just row names to column, and we will name that gene. And then we need to create this ranked list. So within this resdf, just to remind you all, we have these very many columns describing the associated values for each gene. We just want log2 fold change in the gene name. So that's what we're going to do here. So we will use dplyr to filter this data frame and filter for only those two columns. And so we specify dplyr, select, we want the gene, and log2 fold change. And then we want to also do some extra filtering steps, just like remove rows that are NA, as well as make sure there's no double gene values. And then we want to group by gene in case there's multiple ones with the same name. There shouldn't be, but I'm just double checking right now. And then we're summarizing here by the mean log fold change if there are multiple genes. This is likely unnecessary, but I always have this just in case. So that'll create this new data frame called resdf rank, and so you only have two columns, and this is that ranked list I was talking about. And so then we need to deframe this, and this is really just making it no longer a data frame and just creating kind of a list or a text file of the same thing. And then lastly, we're just going to remove any rows that did not have an associated gene ID. Some genes, ensemble gene IDs, don't have a normal name per se, so those aren't going to get assigned a name. That's okay, we'll just filter those out. They typically are not very important genes anyway. As we can see, it only filtered out one line, so not super important. And then now we're going to read in that gene set we downloaded from the msigdb website. And so I'm just going to name this pathway gene ontology, and this pathways. And here I'm just providing the path to that file we downloaded. I just moved that into the folder that I'm currently working in right now. And then here, now that we have our gene set, hold on, before I do that, I will show you what this actually looks like. So you have these 50 gene sets, each with various numbers of genes associated with them. And just to give you an idea, we have hypoxia, mitotic spindle, TGF-beta, so lots of different just biological processes and the associated genes known that enforce these processes. And so then next we're actually going to perform the gene set enrichment analysis. I know, it's so simple, we just really started, but that is how easy this is. And so we're just using the fgsea command, and then I'm just going to feed it that file we just read in, and then we're going to give it the file, or the ranked file that I was talking about earlier, and then we're going to tell it how many permutations are we going to do to create the normalized enrichment scores. Oh. We have to use the one that we filtered for only ones with gene names, so that one line. All right, and so that takes a quick second. And so now we have our GSEA file format here. And then we're going to just start really filtering this so that we can plot it and see what are our top hits. Here we're just arranging it so that the NAS scores are descending, essentially ranking them, or as I like to call, tidying it up. And then we're just going to remove some columns here. There's a lot of extra junk that comes out with this analysis that we don't really need to plot. So we're just going to tidy it up a little bit more. And here we're just essentially leaving out these columns. We don't want the enrichment score because that's not normalized. We tend to only look at the normalized enrichment score. And we get rid of this N more extreme. And then further, we're going to arrange by the P adjusted, so the most significant ones come to the top. And make sure it's a data table so we can properly analyze it down the line. All right, so we're just tidying up our data. And you can actually start to see, once this loads, the associated NES and P adjusted for each gene set here. And this is an ordered list. And so there's 50 gene sets in this, but this is difficult to see when you're plotting it. So I like to just take the top 20 for both. I'll say top hits up. And then maybe fgsea res tidy and just read in the top 20. fgsea res top hits down. And we'll just tail instead of head. Whoop, didn't mean to run that. We'll run it all together once we're done. And then we just need to combine these two to have the 40 most significant gene sets. And we'll just use that rbind command like we used in the previous video. Top hits up, top hits down. Because we didn't run this one. Run that again. There we go. Now we have our top 40 gene sets. And now we're just going to plot a quick ggplot to see which ones are at the top and bottom. Top hits both. And we'll just order it a certain way. And sort by NES score. And we'll just be plotting the NES score as well. Here's just some extra ggplot junk I've used to make it look better. Typically, I like to color the ones that are significant differently than the non-significant ones, so I can actually, at a glance, see which ones are important. These aren't super important. This is just something I do to make sure if I give this data to a colleague, they can actually see what the heck I'm doing, because there's typically no labels. score, and just give it a quick title. I say GSEA hallmarks. And just to make things nice and clean, I always like the theme minimal, but this is really personal choice. Nope. Not that, my bad. Extra F there. All right, let's check and see what our gene sets look like. There we go. All right, now we can see that these are the gene sets that are upregulated in our drug-treated cells. These are the gene sets that are downregulated in our drug-treated cells. And so I'm going to kind of copy the paper that I had referenced in the previous video. This is the paper I was a co-author on. And ultimately, we were focused on the downregulation of G2M checkpoints. So this is looking at a lot of different gene sets, but if you want to focus in on one particular gene set, for instance, G2M, you can actually plot that ranked list chart type thing you see in the initial slide of this video. So I'll kind of show you what that actually means though first. So I'll call this G2M plot. So we're actually going to pull out the data from the ranks file for just the G2M and not look at all of them together. Oh goodness, I have to remember what this is called. G2M checkpoints. Same thing, we're giving it this ranks file. This is just the suggested parameters. And this is just what do the tick size look like, so not super important. I typically use 0.3. And I like to label it just saying plus labs, label, whatever you want to call it, title equals mitotic spindle genes. Alright, let's read that. Hope I typed it all right. Pathways, gene ontology. Oh, I'll copy this then, so it's definitely right. Oh, I clearly typed something wrong. Let's see. I added an S, there's no S. Oh, I have one thing wrong. What did I do here? Oh, I took 2 plus signs, only 1 allowed. All right. Let's see what that looks like. There we go. Okay, so this is kind of that ranked list I'm talking about. So you have your ranked list of genes here, that ranks file, and then the gene sets, the G2M gene set, G2M genes. You can see that a lot of them are clustering here on the negative side of this ranked gene set. And that is why it keeps going down, because it's not really encountering that many G2M genes until it gets to the very bottom of the list. And that's how you get this negative enrichment score. And so that's really actually what we put into the paper. We modified a little bit in Illustrator, but ultimately that was a publication ready figure here. So then next, we're going to try to focus in on the 50 genes that are included in the G2M gene set and plot those and overlay them on the Volcano plot. And the Volcano plot is really just showing every single gene in the RNA sequencing data set for the DEG analysis and showing which are significantly up and which are significantly down. But here we're going to be overlaying these G2M genes in particular. So first, we're going to need this package called ggRepel just so we can label TRP13. And then we are going to create this list of genes from the file we downloaded from MSigDB. I keep typing this wrong. Hallmark G2M check point. Probably typed it wrong. Yes. What did I do wrong this time? I'll copy it from here. I keep putting the S. Alright, so now we have this 200 word list called G2M. We're going to use that to color those points, particularly on the Volcano plot. And so then here we're going to create a new column in our ResDF file, or data frame, called G2M highlight. And we're just going to put the value yes if it's in this list and no if it's not. Just using a quick if-else command. So we're saying if this gene is in this thing we just made called G2M, then put yes. And if not, put no. And we'll come back here. And you can see now we have this new column. And these are all the genes that are in that gene set. And we'll be using that to plot the particular points we want in the Volcano plot. And then last, before we get into plotting, we want to create another column in the ResDF data frame where we say if the gene is significant or not. In the previous video, we had said genes are significant when the log fold change value is greater than or less than 2 and the p-adjusted value is less than 0.05. And so that's really what we're going to do here. Any lines that meet those criteria are going to get a value of up or down. And I'll show you what this looks like when I finish. Thank God it brings it up because I don't want to type all that. And we'll say make that up. And then same deal, but just for the downregulated genes. Honestly, I might just copy this. I have to type it all out. Let's see. Down, make sure this is right. -2. Alright, that should add a new data frame or a new column. So now we have, I have to add one more line. It's not labeling the non-significant ones. And so if it's not either of those criteria that we outlined there, it's going to put non-sig. That's my bad. And so now we have the associated significance values for each gene in the data set. And then we just need to do some quick filtering of this so the plot looks good. Here we're just changing the levels so the points don't overlap. We want the ones that are significant to come on top of the other ones, so that's why we're doing this. If you don't do this, you get the non-significant points on top and it just kind of looks not very good. So we'll say non-sig. When ggplot plots, it plots in sequential order. So it's going to plot the non-significant ones first, then the up, then the down. Anything that comes after goes on top. So that's kind of why we're doing that. And then we need to just order them. Again, reinforcing this plotting hierarchy. The ggplot can be kind of finicky sometimes when you're plotting lots of points. So these are some of the ways that I use to circumvent an ugly plot. And then lastly, just because the paper I was looking at was focused on trip 13. I'm just going to show you while we're here how to filter and label a particular gene you may be interested in. In this case, we were interested in trip 13, so I'm just going to label trip 13 to show you it's actually downregulated. So I'm just extracting that one row from the entire ResDF data frame that has trip 13 in it. So going back to edit this video, I realized I was sitting here typing this ggplot command for about 10 to 15 minutes. So instead of you watching me type that long, I figured I would just paste in the final command and I would just walk you through quickly what each of the rows is actually doing. So really the way the ggplot command works is you tell it what the data you want to actually plot is, and here we're just providing it the ResDF data frame we generated using DESeq2, and we'll be plotting two values for each of the genes in the data. In this case, we will be plotting the log2 fold change, and then we'll be plotting the negative log 10 of the p adjusted value. This negative log 10 transformation inverses the relationship of how you typically think about p-values. So typically the lower the p-value, the more significant. Well, when you take the negative log 10 of these p-values, actually the higher the p-value in this volcano plot, the more significant it is. It's just a more user-friendly way of interpreting p-values. This is pretty much how all volcano plots use this, so just showing you and explaining why we're doing that log transformation. Here we're just plotting all of the points, and then we're going to be coloring them by the value in the significance column. We had added these three values depending on where each row fell in regards to these two columns, either up, down, or non-sig, and so they'll just get a different color depending on what that value is in the significance column. Here we are going to be adding an additional layer of points on top of the original plot, and in this case we're only going to be plotting additional points where the row meets two criteria. The gene has to have yes in this G2M highlight column, which we added right here, and it also has to have down in the significance column, which we added here. So we're really going to be overlaying a second layer of points of only down-regulated G2M genes. And these will be hollow circles as denoted by the shape 1, and so they'll be easy to spot. And you'll see what I mean when we look at the plot. Next here, I said I would label trip 13 within the volcano plot, and the way we do this is we just tell ggplot, hey, we have this other data frame called G1, everything matches up, but we just want to label that particular gene and just adjust it a little bit and make the text a little bigger. And so that's what we're doing here. And similarly, we're just making that same trip 13 point a little larger with the size of 3, so we can actually see which one of those points is trip 13 that we're pointing to. Here, I just like the theme BW. I think it looks the cleanest, but obviously you can change whatever you want. Here, I'm just creating limits for the y-axes and x-axes to actually capture all the points in the data set. Here, I'm just labeling the titles and the axes respectively, and here I'm just changing the size of the text so you can actually read it. This line here is really where I'm coloring the 3 different point types in the significance column. And so remember we had done this leveling where we had non-significant on top, up, and then down. That corresponds to these colors here. So the non-significant will be gray, the up-regulated genes will be blue, and the down-regulated genes will be down. And this is just a trick I use at this leveling to make sure the colors are what I think they are. So if you want to change the colors, or if you have more categories than 3, remember always to do this levels command, and then you know that these will correspond to the levels you created. Here I just like these themes. I think that makes the plot look the cleanest, but again, this is personal preference and you can do whatever you really want here. Last, we have added dashed lines to denote where significant points are. So we've added 2 intercept lines on the x-axes, one at 2 and one at -2. I've explained this before, but those are our thresholds. Anything beyond those, we think is like a large change in expression. And similarly, we have applied this y-intercept line at 1.3. And you may be asking, why 1.3? Well, when you take the negative log 10 of 0.05, it's actually 1.3. And the lower the p-value, the higher that negative log goes. And so this is basically denoting any point above this line had a p-value less than 0.05. And so if it falls, I'll just go to the plot so you can actually see it. And so really, this is the volcano plot, and any of the points that fall in these left or right upper quadrants, those are the ones that are significantly downregulated or significantly upregulated. You can see that I've labeled the colors like I said. Red is down, blue is up, and in these black hollow circles, those are the G2M gene sets, the ones we got from MSigDB, and we've overlaid that on top of the volcano plot. So we can see a lot of the G2M genes are going down. And this explains why we're seeing this very negative enrichment score, because all of the genes are at the bottom of this ranked list. If you imagine the x-axis as kind of this ranked list, a lot of the genes are falling on this side of the rank, hence why we're getting a negative enrichment score. Again, I also said I was labeling TRP13, which I have, and you can apply this to multiple genes. It doesn't have to be one, but in this case, I'm just labeling it as a dark black point, so you can actually see which one of these dots is TRP13. That's pretty much it for the code. The last thing I added was a quick save plot. So here I'm just using ggsave to actually save this image in the folder that I am working. You can change the dimensions, but in this case I'm just saving it as a 7x7, which is the default. Similarly, I added these ggsave functions for these two plots as well. And these will just save a nice PNG image into the folder that you're currently working, and you can input these into your publication or PowerPoint slides or emails, however you want to share your data. But ultimately, these are publication-ready plots, and I just do a little bit tweaking in Illustrator or PowerPoint to actually create them as a final product. So that's pretty much it for me today, and I hope you learned something. Please like, subscribe, and tune in for other Genomify videos. Keep tuned for RNA-seq analysis tutorials, and I'll be expanding soon into ATAC-seq, cut and run, and we'll see what else I have time for. Thanks for joining, and have a good rest of your day.